Today I'm talking with Professor Wenzhen Duan, Johns Hopkins University School of Medicine, where she's a professor of psychiatry and behavioral sciences and director of translational neurobiology laboratory. Dr. Duan, do you want to talk about, we're going to talk today about the potential for these new gene editing technologies for treating neurological disorders, particularly inherited neurological disorders. And you have a lot of focus on one such disorder, Huntington's disease. But before we start talking about that, could you tell me about your background? You grew up in China and went to medical school and graduate school there.
Yeah, sure. It's so nice to meet you, Mark. Thank you for giving this opportunity to chat about the Huntington disease and the recent gene therapy development. Yeah, as Mark already mentioned, I grew up in China and completed my medical school and a PhD degree there. So I really started a postdoc right after my graduation from PhD. For how I get in the medical field, really manifestation followed my parents' advice, who always think doctor is one of the best career and every family needs a doctor. So that's how I get in medical school. But in China, it's like you go to medical school right after high school. So I got into the medical school when I was 16. So I kind of jumped a couple grades in elementary school. So I went to the medical school earlier than average age. But in that time, I was so brave. I thought, oh, after 5 years medical school, I'm going to cure any diseases and help my parents and everybody around. But as more I learned, I less confident I can reach that ambitious goal when I get into the medical school, especially when I learning about the neurological disease. I found there's almost no cure for majority or basically all neurological diseases, no cure. All the treatment, especially that time, is very symptomatic relief, but none of the treatment are going to really either delay the onset or slow the progression. So I decided right after medical school, I decided instead of practice medicine, I decided to go to a research track, which in China, I have to finish like 3 years master program, another 3 years PhD program. So total is like here, like 6 years graduate schools. And I started neuropharmacology because I think that's the way to develop a treatment for like disease. So my master research is really focused on develop some medicines from Chinese medicine. One of the example is called quercetin. So it's like more like antioxidant, have more like other anti-aging probably properties. So really studied this medicine in stroke models. And then I move on to my PhD. Actually, I switched to a different school. I finished my medical school and a master in called Shanxi Medical University, which located in northern China, about 300 miles away from Beijing. And then I moved to a PhD program in Peking Union Medical University. There I did 3 years continuously neuropharmacology, but really focus on develop treatment for Alzheimer's disease and aging. That was my background in China. And then right before I finished my PhD that time, so majority of the people come to US. Yeah.
Okay.
Yeah, and then right before I finished my PhD degree, that time in China, most of the PhD graduates come to US or Western country to do the research because we have much advanced technology and equipment resource here. So I just decided to come to United States to continue my postdoctoral research. I found ad from a journal called Neuro, and Dr. Madsen was recruiting a couple of postdoc focus look at how apoptosis is involved in neurodegeneration. That time was like really the new advanced field, how the cells die in a neurological disease. So get me really interested in this field. So I contacted Dr. Madsen. I think after a couple of days, he offered me the position. That was like fast track. So I came to United States, work with Mark at the University of Kentucky for first couple of years. And really with advice from Mark, I became interested in Huntington's disease in that time, because that's one of the most common hereditary neurological disease from like one gene caused disease. And Mark was interested in studying how diet, lifestyle affect the neurodegeneration. So I started those research in the Huntington's disease and continue on for more than 20 years now. Yeah.
Yeah. So back then in the late 1990s, so it was possible to recruit like the top students from China. So when I recruited you, right, you're a top student. And so this was really valuable for United States research because we're getting, like we call the cream of the crop from China. And then it's changed though now.
Yeah,
absolutely. Now it used to be, and you're an example that, and I had quite a few postdocs from China that they wouldn't even consider really going back to China. But now I've had a few postdocs that have gone back to China because China has put in the last 20 years a lot of money into research and development. So I think a lot of people know this. China is going to be ahead of the United States in science and technology pretty quickly if we don't wake up here and devote more money.
Yeah, that's so true. I think in my time, we came here, we think we will spend the rest of life here because the research environment, you just cannot compete. When I'm doing my PhD, I often have to buy like a reagent from Sigma. This really have to be imported. Like you put one order, you have to wait for a month to come. But nowadays, there's so much advancement and investment in the science technology in China. So the research environments totally changed compared to my time like 20 more years ago. Yeah, lots of young fellows, after they finish postdoc training, they went back to China because they can do identical research as we're doing here. Yeah.
Okay, so tell us about Huntington's disease. What causes Huntington's disease?
Yeah, Huntington disease is one of this we call autosomal dominant inherited neurodegenerative diseases. It's one of most common in a monogenic or inherited disease, inherited neurological disease. So it's caused by this gene also called huntingtin, which is in the Huntington gene exon 1 regions. They have CAG trinucleotide repeats in the normal Huntington genes. But once this repeats extend more than 36, so the carriers would have high risk to develop Huntington disease. Once the research reach to 40 repeat, it's like 100% the carrier will develop Huntington disease during their lifespans.
So the CAG, that 3 base sequence in the DNA encodes for 1 amino acid, which is glutamine. Mm-hmm. So there's this abnormal stretch in the Huntington gene, or in the protein product, where you've got all these glutamines in a row at an abnormal amount.
Yeah, exactly. So the CAG codes for glutamine. So also that's why Huntington disease also we call polyglutamine disease. Yeah, you have expanded glutamine in the protein like N-terminal. Huntington is a huge protein. They have 67 exons. So this CAG repeat is in the first exons. So it's very coding the protein, the very N-terminal of the protein. Yeah. So with this like 40 repeats, we call full penetration. That means they're 100% going to develop disease. And their expansion of this CAG is negatively correlated with age of onset. So patients often have longer repeat, going to develop disease earlier. But however, the CAG repeats cannot fully explain the onset of disease. So they only explain about the 60 to 70% onset of disease. And a large genome-wise study showing other genes we call Huntington disease modifiers, and also kind of modulate the onset of disease, which we can talk about later when we mention about the gene therapy new targets. Yeah, it's very, very devastating disease. Once the disease start, it's about 15 to 20 years, patient will be totally
And so someone that either from their mother or father, they inherit this abnormal Huntington gene, then they have in their genome, they have one good normal copy of the Huntington gene and one abnormal, and they develop disease. So this is typically referred to as gain of function. In other words, they still have one normal gene, but the abnormal gene is doing something that we don't fully understand yet, right?
Right. So this is a typically we call autosomal dominant. So you just need one allele with mutation to develop the disease. So as as Mark already mentioned, so most 99% patient we call heterozygotes, so which they have one normal allele and one mutant allele. Very, very few case they have like both allele, which has to be both parents have one mutant allele. They just need one allele to develop the disease. And the clinically, they have very affect almost every, you can think about almost every functions in the brain. So they affect the motor functions, they affect like mental status, and they affect the cognitive dysfunctions. So the patient can start with any kind of, like lots of patients, maybe 40% patients start with depressions or sleeping problems, not really motor, but the clinical diagnostic right now still based on the motor symptoms. Once the patient have motor symptoms and they can diagnose, oh, this is onset of the Huntington's disease. But this disease is very interesting. Like many other neurological diseases, disease started much earlier than the clinical onset or clinical diagnostic, maybe like even 20 or more than 20 years before they develop the clinical onset. If you use this technology called magnetic resonance or imaging, look at the brain of the Huntington disease carriers. So the brain already lost, like some lost 50, some even lost 70% this brain region called caudate putamen, but the patient still perform seemingly normal functions. It's a very interesting disease. Think about how they develop, how they clinically diagnostic. This is very important for future considering treatment when you suppose start to treat the disease.
And so this brain region that's affected earliest, you mentioned caudate putamen, or we refer to as striatum, basal ganglia. People should know that there's motor neurons in your cortex that's their axons go down to your spinal cord and form synapses on what are called the motor neurons that innervate your muscles. Okay, and those motor neurons in your brain, the upper motor neurons, they're constantly active. They're constantly sending signals. And so the basal ganglia that's affected early in Huntington's disease, the function of those neurons is to inhibit unwanted movements. So actually, that's kind of a key feature of Huntington's. They have this constant moving.
Dancing syndrome called,
So this pathway from the cortex to your muscles isn't being controlled.
That's good to maybe give a little bit of background about why this patient started from this dancing or career symptoms. So as Mark already mentioned, in our brain cortex, there are specific neuron called the pyramidal neurons. They project into the spinal cord or peripheral, but also a group of pyramidal neurons, they project to the striatum areas. And that's very, very interesting is like the most vulnerable neurons in the striatum in Huntington disease called the medium spinal neurons. So this group of neurons project to two different pathways. One we call indirect pathway. So the medium spinal neurons project to the called external segment of the globus pallidus. So this pathway we call indirect because they indirectly control how your brain want to your muscle act. So usually this indirect pathways suppress the movement. So once this pathways is impaired in Huntington disease, the patient cannot control their movement. That's where we also call dancing syndrome or chorea in the earlier or middle stage of the Huntington patient. But another pathway we call direct pathway, which is a medium spinal neurons projected to internal segment of the globus pallidus. So this, and also substantia nigra. So this pathway is really modulate how you move. So it's really in the later stage of the Huntington patient, it's really the direct pathway is impaired. That's why the patient start from chorea or uncontrollable movement become hypokinetic or very slow movement in the later stage. So these two pathways, also these two pathway neurons in these two pathway controlled by different receptor, like indirect pathway neurons express D2 dopamine 2 receptors, and the direct pathway express dopamine 1 receptors. But the really molecular mechanism or cellular mechanism, how this impaired in different stage Huntington disease is not completely understand. Yeah.
And these vulnerable, you call medium spiny neurons, they deploy the inhibitory neurotransmitter GABA.
Right. Yeah,
Right.
they are GABAergic neurons. Yeah.
Okay. Now, of course, once the gene, Huntington gene, was identified and associated with Huntington's disease, and then it became possible to genotype people, that is, to have their DNA sequenced. And so, for example, if you're the child of, and one of your parents has Huntington's disease, you have 50% chance of having the mutant gene, and you can know that if you want to.
Exactly.
And this becomes important when we get to gene therapy, I think.
Mm-hmm. Yeah.
Okay, so what we're getting closer to the gene editing as a potential treatment for either actually preventing or treating Huntington's disease. But what did you learn about Huntington's disease, before you got to using these new CRISPR gene editing technologies?
Right. So the Huntington disease was discovered like more than 200 years ago, but the gene responsible for the disease, Huntington mutation, was really discovered in 1993 by a large group of the geneticists. So since the discovery of the mutation of the Huntington, and their animal models, cell models start to establish. So we learned so much since 1993 to the first decades, basically from 1993 to the earlier 2000. So we just focus on understand how this mutant Huntington genes kill the neurons, right? So we use different model system. But it's interesting is you almost every signaling pathway you can think about the critical for cell survival is affected by this mutant genes. Mitochondrial dysfunction, transcription inhibition, and axonal transportation, and some inflammatory pathway. So almost like it's hard. So the more we learn about the mutant Huntington doing to the cells, the more difficult we think targeting to each signaling pathway develop as a therapeutics, because if you hit one pathway, maybe another pathway is still going on, such as like BDNF transcription was inhibited by mutant Huntington, some like BDNF mimetics or TrkB receptor agonist has been investigated in a different lab, including my labs. But the more and more we learn, find if we don't target the root of this disease or the disease at its origin, it's hard to completely get rid of the disease. Yeah. So that's why for past 2 decades, with this gene therapy technology is advanced, people shift more therapy of the Huntington disease to we call Huntington lowering or the Huntington targeting therapies, which include multiple target. You can target the DNA levels, RNA levels, and the protein levels. Yeah.
And so most of our viewers and listeners have heard of CRISPR, which is a hot topic now in biomedical sciences. And can you try to explain what is CRISPR?
Yeah, CRISPR, the full name is called clustered regularly interspaced short palindromic repeats. So it's long name. I think easy for the viewers to just call CRISPR because everybody heard about the CRISPR now probably. So and CRISPR is more like if I use lay language, it's more like mission target. So this system was discovered in the bacteria when bacteria fight to exogenous like virus infection or other outside pathogens. So the bacteria adapt to this specific sequence in their genome. So when next time virus or other outside pathogen invented the bacteria, so they will recognize it and then they eliminate it. So the CRISPR is more like a specific sequence you can design, which we call the guiding RNA, to target your gene-specific sequence, and then associated with this enzyme called CRISPR-associated system, which is a DNA nucleus, so they can cleave the DNA. So it's more like you have this biological missile to target the genes you want, and then you associate with the kind of like a molecular scissor, you cut or delete or replace the mutation or unwanted sequence in your genome. So since actually last week was the 10 years anniversary of this technology, so the CRISPR-Cas technology for gene editing won the Nobel Prize in the 2020 by 2 female scientists. So that was exciting. In the first paper came in 2012 about this new gene editing technology, but within 10 years, like we already some clinical trials for the different disease already going on. So it's very effective and promising gene editing technology, especially for the genetic disease. I think brings so much hope because they are relatively cheap and can target more specific genes, and it's very efficiently so far as we know compared to other like zinc finger binding proteins, talons. But each of those gene therapies have their like pros and cons, so we can discuss a little bit later. But this technology, I think, brings so much hope to the monogenic or other genetic diseases.
Okay. So, right. So that you said what CRISPR stands for, and in that is palindromic repeat. So a palindrome is a sequence that's identical in both the forward and reverse directions. So it's a DNA sequence. So do our genomes have these sequences?
In our genomes, they are those sequences. That's how we identify those sequences and specifically target with the guiding RNA.
Right.
Yeah.
Right. So in theory, then, this enzyme that's coupled to the RNA can be designed to go to any gene in the genome and do specific things to that gene. So Huntington's is a little more complicated than I think some other inherited neurological disorders because, so for example, as you know, my lab's done a lot of work on Alzheimer's disease, Parkinson's disease, and they're somewhat rare early onset forms of Alzheimer's and Parkinson's that are inherited. And some of those, as with Huntington's, are inherited in autosomal dominant manner. So if you have the mutant gene, you will get the disease. But in most cases, with Alzheimer's and Parkinson's, it's just one amino acid change
Right, point
in
mutations.
the protein, and so that seems to me with this CRISPR technology, it might be easier to target that. Well, then there's another issue that comes up is in the case of Huntington, mutant Huntington, one approach would be to, and the other disorder, to selectively delete or inactivate the mutated gene. And then the person will still have one copy of the normal gene. And in some cases, they may be able to function okay with one copy. So anyway, do you want to describe your approach to Huntington's? Because you've got these CAG repeats, right?
Right.
Now, is your approach to correct the mutant gene or to inactivate it?
Yeah, I think that's really good point. As you mentioned, it's really very challenging for Huntington because normal Huntington, you also have CAG repeat, it's just a little bit shorter.
Yeah.
So that's why it's very challenging to target only mutant allele if we target the CAG regions, right? Because the normal also have CAG, they're just short, usually about 17 to 20 CAG size. But however, I think for the Huntington disease, to answer your questions, I think the best way is to eliminate the mutant genes. It's hard to correct to the normal size, but another biological phenomenon called the somatic expansion. So the DNA undergoing like the damage and repair. So why every cells in our body express Huntington. So for the Huntington disease patient, also every cells in their body express mutant Huntington. But however, why only these medium neurons specifically died, right? So one possibility is people found when they do the sequence of the single cell RNA-seq, they found that this medium spinal neuron have thousands CAG repeats. Even your diagnostic from drop of blood, you have 45, right? Average is like between 40 to 50 the patient. But this medium spinal neuron have thousands repeats. So those repeats came from, we call the DNA like damage and repair, we call it somatic expansion. So that's another mechanism.
The mutation is getting worse
getting and worse in certain cells. Yeah, that's how they probably killed medium spinal neurons first. We don't know the mechanism, but that's one of the phenomena observed. So it's really hard to correct because they're going to be during the cell like division or from the neurons is post-mitotic, but they are constantly under DNA repair and damage and repair during the lifespan of the neurons. So those can becomes longer and longer. So ideally, we want to really get rid of the mutant genes because they seem to don't have any good functions. One normal allele probably can play most Huntington's endogenous functions. Yeah. But regarding the CRISPR-Cas technology for Huntington disease, there are two categories. One we call non-allele specific, which we targeted to the CAG regions. So you eliminate both or reduce both mutant and wild type Huntington. So that's based on this big clinical trial called antisense oligonucleotide. They targeted the pre-message RNA, but they couldn't distinguish normal versus mutant. So they both reduce both allele of the mutant and wild type Huntington resulted in the protein expressions. So those has been actually the first clinical trial for the Huntington lowering therapies has to go through phase 1, 2, and 3. But unfortunately, the clinical trial was suspended in the 2021 March last year because there are some unwanted side effects in fact start develop in the patient, and we still want to hear what's really the reason cause this stop this clinical trial. But several potential possibilities. One is because we target mutant and normal Huntington. Basically, in the animal studies, if you reduce 40 to 50% wild type Huntington, this seems okay for animals like 1 year, but who knows for the human, like how much lower, it's very safe. Another possibility is the dosage, probably because they studied low dose and high dose, and the high dose group seems to have very strong side effect. Another is those treatments have to go really invasive injection, they call it intrathecal. So basically you do the spine puncture every few weeks. So that can also cause lots of consequence. So we don't know yet, but those data should be available soon. So we will learn how this antisense oligonucleotide trial failed in the first, but this is one of the largest clinical trial. But regarding the CRISPRs, we can target the CAG region, which is non-allele specific, which my lab also did a little bit of study in the animal models. But another possible way called allele specific, because in our genome, there are this called the single nuclear polymorphism. So in the mutant Huntington genes, there are 60 to 70% patients carry this different single nuclear polymorphism. So we can design the guiding eye targeting to this specific, we call SMP. So that can result in allele specific, but that doesn't cover all Huntington patients, maybe only 60 to 70% of the patient, yeah.
And yeah, this is another big issue in trying to treat neurodegenerative disorders, is that once the person becomes symptomatic, there's already a lot of damage done.
Yeah.
And so in Huntington's, Alzheimer's, Parkinson's, the inherited forms, ideally, one would have some treatment that could be administered before they even become symptomatic, and a person can know whether they have the mutant gene.
Yeah.
And so I want to digress just a little bit, but along these lines of, prevention and going way back.
Yeah.
So if I was a child, and this is me from a scientist's point of view, if I was a child who had a parent with Huntington's disease, I would want to know if I had the mutant gene as soon as possible. And that would allow me to if I get married, want to have children, then there are ways that I could ensure that I don't have a child with Huntington's disease.
Yeah.
The one is don't have any children and adopt. That's one approach. Another approach, which the technology has been around for quite a while now, it's called in vitro fertilization and embryo selection.
Mm-hmm.
So the way that works is, people have heard of in vitro fertilization for women who have trouble getting pregnant. So that's the same part. You essentially fertilize the wife's egg with the man's sperm, and then eggs will be, some of them will be fertilized, and they'll start dividing in the embryos. And then now it's possible to, in the embryo that only has like 8 cells maybe, or 16, to take out one cell from the embryo,
mm-hmm.
and you can genotype it.
Right.
And you can say, okay, if we take this embryo and implant it in the woman, it's not going to have Huntington's disease. So that is possible now. So I guess my point is, in theory, it's possible within one generation to eliminate all dominantly inherited autosomal disease. Now, there's a lot of ethical issues, and this is like a personal decisions of each person, but I'm just saying in theory that's possible. But I don't see that happening anytime soon, mainly because of, I don't know, what are your thoughts on that?
Yeah, I think this is definitely one of the options for people who inherited this disease genes. But my understanding from the field, like about half, 50% people don't want to know whether they have the genes because
Yeah.
they couldn't just face to this, like every day I think about the wake up, I have the Huntington disease. But the other 15% patient carriers, yeah, want to know as early as possible. In US, once they reach age of 18, so if you're one of the parents have the disease, you can go to the Huntington clinic, get drop of blood, do the PCR, and then you will know whether you inherited these genes or not. In terms of the future, like next offspring, there are options use IVF to pre-select embryos to get a disease. Some people are doing that, but there's some ethical issues. But another aspect to consider is, as I just mentioned in the very beginning of this chat, so the patient, the CAG repeat threshold to lead to the Huntington disease, it's 36. From 36 to 40, this we call that you have high risk develop Huntington disease. Some of these people with this repeat might not never develop disease in their lifespan.
Oh.
So those people will not know whether they have Huntington. So this is the part of the carriers we probably never eliminated. Also, seems as a generation pass on, and at least from a parental side, seems CAG getting longer and longer. So that's another possibility. Even some people said that they have between 29 to 36, we call intermediate. So those carriers probably as many generation or few generation, their offspring might have the mutant.
So there's some variability in the age of onset of disease, that is, what age the person starts to have symptoms.
Right.
But what's the range of ages?
Yeah, the average range from the repeat, we call the adult onset, is repeat around between 40 to 50. The average onset is in the middle age, it's age of 40 to 50. That's very average. If you repeat more than 50, like 60 or even longer, they can start in the juvenile form. So we call juvenile Huntington's disease. So basically they average start middle age, but can really start at any age. Some start really late. So the CAG explains 60, 70% after disease onset, but the other modifiers, if two persons all carry this mutant Huntington genes, one have different modifiers, can start really late, and another one can start really earlier. So it's very lots of variabilities regarding the onset of disease. Yeah.
Interesting. So Wenzhen, can you tell us what gene therapy did you do in the Huntington mutant mice? So you have these mice with the mutant gene. Human Huntington gene, and they develop some problems controlling body movements, and you've done a lot of work doing imaging of their brains. I guess, can you kind of summarize what you're looking at in terms of the behavior of the mice and changes in the brain, and in looking at how the CRISPR-based gene therapy is affecting the disease?
Right, yeah. So with this genes was discovered, so the first few mouse models generated, we call transgenic mouse models, which is mouse overexpress either partially or full-less of the mutant Huntington genes from humans. So those mice develop some motor symptoms, some cognitive symptoms, and they have the brain atrophy. Similarly, as the patient, they have also this pathology biomarker called mutant Huntington aggregations. Along with this genetic models advanced, now we must switch to we call knocking models. So because you don't overexpress proteins, right? When you overexpress any proteins, even normal proteins might have some adverse effect, not purely from the gene mutation. So now we must switch to knocking, which we knock in piece of the human Huntington, basically is the axon 1 region with expanded repeat into the mouse Huntington homolog. So in this case, mouse have one normal Huntington, but one allele with the mutant Huntington. So from genetic aspect, so this is really mimics the disease. So those mouse models also develop a similar like motor problem, some anxiety and cognitive, and also have very selective brain atrophy. They saw this striatum atrophy occurred earlier stage and gradually expand to other brain regions. So one interesting point that Mark just mentioned, since Huntington disease carriers, if they want to know, they can know way before they have clinical onset, right? So that's one of the advantage for treatment development, because many neurological disease like Alzheimer's disease, Parkinson disease, without the family genetic cause, we don't really know when the disease is going to start. We only can treat the disease when they start, but often it's very late. No matter what this neurologic disease, when they have clinical symptom, maybe 30–50% of neurons in specific brain region already lost. So that's very challenging for any treatment to really rescue or delay or slow down the progression of the disease. So for Huntington disease, is advantages. We will know these genes, whether the patient have the genes way before they have the clinical symptoms. So that's also one of my equally important research area in my lab. Besides develop treatment, invest a lot of called biomarker studies. So we try to use non-invasive like MRI approach to look at when this Huntington brain start functional change way before they lost striatum or any other neurons. So we hope this earlier functional change can give us an indication. Maybe we can start the treatment much earlier in the stage before they start the disease. So the goal is we can completely really prevent the onset of disease. So that's our like very idea goal. So we use this magnetic resonance imaging technique. We can look at not only structure change, we can look at blood flow changes, which reflect the metabolic change. We also can look at like glucose metabolism recently with this glucose test. So it's very powerful non-invasive MRI approach to look at when the brain start have some abnormal function change, but the patient will not start clinical manifestation.
Yeah. And right, so you have these Huntington mutant mice, they develop motor problems, their striatum gets smaller. And so how did you get this CRISPR Cas9 system, essentially enzyme that's targeting a specific region of the Huntington gene? How did you get that into the mice?
Yeah, so this is one of we call the adeno-associated virus as a delivery tools. So we put this CRISPR-Cas and the guiding RNA in the AAV, abbreviation for this delivery tool called AAV. So we put this CRISPR and Cas9 in a separate AAV, and then we co-injected in the mouse models, we co-injected this brain region called the striatum, which is most vulnerable. So we injected this in the striatum, just one injection, and then along with time, first we want to confirm whether this CRISPR-Cas really knocking down or lower the Huntington levels. So after about 1 month, we sacrifice mice, we dissect the striatum, we look at the Huntington levels, which include both mutant and wild type Huntington levels. We did see lower both mutant and wild type Huntington levels. And along with time going on, we use mRNA or motor function or pathological pathological analysis, look at how we call efficacy, how this treatment is going to improve in the mouse models. So we did see improvement use this technology because we only target in the striatum, which is the most vulnerable region. And then we cause about a 30–40% reduction of the wild type Huntington, which not cause dramatic adverse effect. At least in the mouse model in this stage, we think this is one of the promising for further look at the safety, of course, efficacy in the large animals, maybe before we move to the human trials.
And this virus, AAV virus, it's an innocuous modified form of the virus that's been used. These have been used in humans.
Humans, yeah.
Quite a few studies. Now, my understanding, the CRISPR method has been, there's been two that I'm aware of, clinical studies. One was in in this,
One an eye disease,
yeah, eye disorder. It's called Leber's
Mhm.
syndrome, where the person essentially becomes blind.
Blind, yeah.
And it looked like in this initial patient, maybe there's some benefit.
Yeah, it's promising.
And then the other was sickle cell anemia.
Hmm.
And that person, I looked at the, and she had a remarkable benefit from targeting the genetic problem in sickle cell, where the hemoglobin isn't being produced, I guess.
Yeah, for this peripheral disease, I think CRISPR is more powerful because they can take the cells out, engineered, put it back, right? So you don't have this called blood-brain barrier problems.
Yeah.
For the eye disease, also they can deliver relatively easier than other neurological, which you need intra-brain deliveries. So delivery always a big challenge for any gene therapies, include CRISPR, like zinc finger binding proteins, and also like talent. Any of these will face challenge for delivery, but the AAV is one of relatively safe, effective, because AAV not integrated to the human genome, so that's relatively safe. They don't cause much like severe immunogenicity, but people still worry about for neurological disease, you need maybe 30, 40 years, right? It's Huntington, if you give them way before start the disease, like 20, 30, they need like many years. So we don't really know with that extended lifetime whether this still become immunogenicity, still become a concern. But another powerful delivery agent called the lipid nanoparticles, which is very powerful. People who these 2 years, maybe everybody know lipid nanoparticles because to this COVID vaccines, like both delivered by lipid nanoparticles.
Yeah.
But that's all the peripheral delivery. Whatever you go to the brain is always challenging. Also for CRISPR-Cas, you want to just one straight delivery. You don't want to repeat. Once you repeat, the Cas9 might become resistant, so it might cause other problem. But that's the one of the direction. My lab actually is just very initiative study with this professor in the school engineer. We try to deliver the lipid nanoparticle can either through the intravenous or intranasal delivery some maybe CRISPR-Cas RNA into the brain, because hopefully they will have more broad brain delivery. Although the Huntington disease most affected the striatum, but the other brain region also affected. So we want to knock out the mutant Huntington as many cells as possible. So that's another direction, is really in the research.
Yeah, the intranasal delivery is interesting because I guess easy way to explain it, there's less of a blood-brain barrier.
Yeah, so the area.
Intranasal. And that's actually why, certain people who are drug abusers, like cocaine, for example, they
sniff,
because it goes right to the brain. And even big molecules like proteins can get into the brain. There's been trials of intranasal insulin in Alzheimer's disease, for example. So these small, the size of these lipid nanoparticles, we used to call them liposomes.
Right.
So there's the word nano at some point during my lifetime, maybe 20 years ago, I think it was when we were at Kentucky, this nanotechnology term took off, and then everybody to make it sound more sexy, I guess they put nano into it. So anyway, but they're small. What's the diameter of a liposome, do you know?
So it can vary. Like ideally for bread delivery, we want them under 200 nanometer, the diameter. But even it's a little between, for example, between 200 to 300, according to my collaborators, that they can engineer some specific genes or vectors in the surface of lipid nanoparticles, help them kind of pass through the blood barrier. So one is the size, one is really they can engineer multiple different components in the surface of the liposomes, so make them more easy to pass
Huh.
blood-brain barriers. So
Okay, anyway, and what do you know about other, we call preclinical trials of using CRISPR in neurological disorders? I see there have been some studies in mouse models of Alzheimer's.
yeah, Alzheimer's published a couple years in the Nature Neuroscience. I think that was first CRISPR targeted to the APP, right?
Yeah.
And then some others like Parkinson's target to alpha-synuclein,
Yeah.
but the most CRISPR targeted to the this genetic caused pure genetic disease basically is the way to go. And also besides CRISPR-Cas9 is another class which we recently actually completed a study and under review in the Nature Neuroscience. It's called the CRISPR-Cas13D families. Instead of target DNAs, those enzymes targeting RNAs, which is maybe less ethical in the future because you don't edit in genome, but you also prevent your RNA produce proteins. So we just complete a study in the mouse model and the human iPS cells uses CRISPR-Cas13D. We found by select a few different guiding areas, we were able to knocking down only mutant Huntington RNA and proteins in the mouse models humans. It's very exciting. And yeah, we hopefully we just submit our revision a few weeks ago. Hopefully our paper going to accept it so people will know this is a new CRISPR-Cas.
And when you're targeting the messenger RNA, in this case, the messenger RNA that encodes the abnormal protein. Huntington, with the CRISPR-Cas13 system, will you get constant suppression of that mRNA, or is it going to be transient?
Yeah, we use the AAV delivery.
So it's continually.
Right. It's continuously, yeah.
Oh, that's very exciting.
Yeah, it's very exciting. Beginning, we just screened it in the human cells, and then we found this. We don't know the reason, but it's very we target to the CAG region with different starting codon, and we found a couple of them guiding are just effectively knocking down only mutant, and then we screening in the human iPS cells, which is collaborated with my professor in UCSD. They are really the RNA labs. And my labs did all the mouse studies. We are very excited to see how this can expand to like long-term or other models of the disease. So that's another potential gene therapy. And also the Zinc finger binding protein approach. This is allele specific regarding in the Huntington's disease because you can engineer the DNA binding regions to only binding to the expanded CAG. So this has been done all in a preclinical model of the Huntington disease. But right now, a couple companies try to develop effective, less adverse delivery approach to the larger animals before we can move to the human. So another one is like recently a couple companies found these small molecules, which you can already take past blood-brain barriers, but they regulate called the gene splicing and also can lower the Huntington levels. One is really from a serendipity finding. The compound called branaplam was developed for the spinal muscular atrophy, but they found that in their patient samples after the treatment, the Huntington levels was lower. So they moved to the Huntington disease model. They found this is really dramatic lower Huntington, both mutant allele and normal allele. So they now initiated the phase 3 clinical efficacy trial in the Huntington patient. Another is a small molecules through the big screening from this company called PTC Therapeutics. They purely just screening any compounds can lower the Huntington levels. They also had one candidate they prepare for clinical trials. So like small molecules are possible as also regulate the Huntington levels besides this gene therapy. So if those clinical trial pass, that will be much faster move to the human because they are like orally give, you don't have much other AAV problems. So it's very promising.
The NIH has a a program for rare genetic disorders to accelerate treatments. In other words, they aren't as strict in terms of patients being able to try some new treatment that hasn't gone through like the rigorous testing. And yeah, I think that's a good program. And I guess I should mention that, and I'm gonna have a podcast with on pain, right, with Steve Waxman at Yale University, and there are some rare genetic disorders that result in kind of hypersensitivity to pain. And I actually have a mutation in a gene that encodes a voltage-dependent sodium channel. It's a protein in the membrane of neurons that convey pain that the sodium ions move through it. And the mutant channel I have it opens more easily. And so the neurons that convey pain are more easily activated.
Yeah.
So I've actually I'm going to have a meeting with some people at the NIH rare disorders thing to talk about.
Yeah, this is a special track called the rare orphan disease designation.
Yeah.
So really the fast track, and also you don't need that large patient population. You can do like 30 patients, 20 patients. Yeah.
And so in the case of this kind of disorder, the neurons that would be targeted are right next to the spinal cord in what's called dorsal root ganglion. And so with that, in theory, it's actually maybe a little easier than the brain, because you could actually have the CRISPR Cas9 targeting sodium channel in AAV and
inject
it into the dorsal root ganglion.
Right. You don't have the blood-brain barrier problem. Right. Yeah, that's much easier compared to this brain.
Yeah, and that's just like an outpatient thing. So.
And also with AAV, you just like one injection work for like many years.
Yeah,
20 years. So
yeah, you just have to get it where the neuron cell bodies are.
Into the perfect place.
Right. So there may be some applications of the peripheral nervous system disorders in theory that could, yeah, and then you mentioned delivery. So one approach is to inject the AAV with the CRISPR Cas9 system into the spinal fluid. There's that can be done, at the level of the spinal cord. The same procedure used to remove spinal tap. You essentially you do a spinal tap and inject something.
Right.
And then that AAV is gonna, for example, in ALS, right? Motor neuron disease, it would be bathing the motor neurons. So there are actually some preclinical studies in animal models of ALS with CRISPR-Cas9 and some of the
to the SOD1 mutation. Actually, for the ALS, it's a little bit easier because all the motor neurons in the cortex. So if we inject anything from spinal fluid, so the most area they penetrate is cortex.
Yeah.
For the Huntington, a little bit because the basal ganglia is a little bit deeper in the brain. So that's why we do the actually one of the first gene therapy clinical trial use AAV started end of last year by this company called Unicure. So they use AAV5 delivered the RNA interface called the microRNA into the stratum of the patient. So Hopkins is one of the center. I think first they completed the first 10 patients for the injection. So far, seems they don't have much safety concern. So they will continue to do another. I think the total is 26 patients for this phase 1/2A safety trials. So that's also very exciting because you just need one injection. For the lumbar one, so you need repeatedly every 8 weeks or every few weeks. So that's kind of very invasive. Yeah, so this gene therapy is really advanced in the last decade. I think provide much hope for this genetic disease.
Yeah. And I should also mention that there's other uses for CRISPR Cas9 system. A lot of people are using this way to not to correct a gene defect, but to introduce a gene defect. So to make, for example, mouse models of some disease, it's a lot quicker. It's a lot quicker than the old way.
Right, the people make knocking and knock out the mice within a few weeks. Yeah.
Yeah.
It's very commonly used for genetic animal models these days. And also I want to mention, beside this gene therapy directly target Huntington, there's another pathway, the DNA repairs. I think it's very promising. One of the clinical trial target this gene called MSH3, which is a mutant homolog 3, one of the key enzymes involved in the DNA repair. So they found if they knocking down this MSH3 in animal models, they can prevent the somatic expansion of the mutant Huntington, which can really rescue some of the phenotypes in the animal models. Yeah.
But are they impairing DNA repair?
They really inhibit one of the component in MSH3, which they have to probably, the long ways wondering if you really impair this DNA repair with cancer or other problem. But so far, since the HD Huntington disease is very fatal, so they allow them to start knocking down the MSH3 in Huntington disease, which can prevent this CAG repeat expansion. One of the major maybe cause the medium spinal neural die. Yeah.
Interesting.
Yeah, they started clinical trial very fast. Yeah, lots of new trials started in past decade. Yeah.
Okay, and I guess we could mention your the clinical program on Huntington's disease at Hopkins is Chris Ross is the head. Is that right?
Yeah, Dr. Chris Ross, which is my mentor, he's a medical doctor and also PhD. So he's leading several different clinical trials going on. One of the uniQures microRNA introduced by the AAV, we are one of the side. So Chris is conducting this kind of clinical trial. And for the antisense oligonucleotide trials, we also one of the larger side to test this in the clinical patient. And also he's leading some clinical biomarker studies, also use a similar MR approach we used in the mouse models. Those approach can also apply to the human brains. So this is really one of the beauty in Hopkins. We have this preclinical and the clinical right next to you. So we can really translate the finding from the bench to the clinical, which is my goal in medical research. You can really help the patient. And
so now are you thinking of trying to work with clinicians and the various regulatory people to get a trial of the CRISPR Cas13 or?
Yeah, that will be a long way to go. We have to do, this is the first study we did in the mouse models. Another group just published recently in the Science Advances. They also use a similar approach, found they can knocking down Huntington in one of the transgenic mouse models, but they didn't do much efficacy. So, but I think this is promising because they're target only mutant allele. So my goal is to continuously look at the long-term safety, long-term efficacy, in different models, including like human iPS cell-derived organoid models. So really see within the disease range, because one of the things I missed is for most animal models, we have this huge expansion CAG. If you put 40, 50 repeat in a mouse, they don't develop disease. Yeah, mouse somehow just resistant to the mutant genes. So we have to artificially expand the CAG to hundreds, 200 in order to have the mouse have manifestation of Huntington disease. But now they have this human iPS cell derived model, which is a human CAG size, most around 40, 50. So actually one of the critics from reviews said, you beautifully saved the Huntington mice. We're not surprised because you have 200 versus 7 repeats. It's very easy to distinguish. But when you move to the human, they have 20 versus 40. Your approach still able to distinguish between mutants? So actually, we did the human fibroblast cells with 46 and 55 repeat, with another is 17 to 20 repeat. Yeah, with this CRISPR-Cas13, they can still selectively only knocking down mutant allele. So that's kind of part very exciting for us. Yeah, we want to really apply for NIH funding to let us to do like long-term efficacy in animal models, also in the human iPS cell-derived organoid models. If we can achieve allele specific in the human CAG, repeat in the human cell model, organoid models, that would be really promising for us to move to the clinical trial in the future. Yeah.
Great. Well, we've covered a lot of ground. You're at the cutting edge of this research, and yeah, the next few years and certainly decades are going to reveal a lot of the potential of this new gene editing technology, and also, raise some ethical issues, which NIH is still kind of wrestling with, I think, the details of what kinds of things. And then this unfortunate situation in China, right, that caused a lot of concern, where there was an investigator who apparently without approval did some editing in human embryos, I guess.
Right.
Yeah, so, but yeah, I think overall this is really exciting, and it's going to move forward.
Yeah, definitely it's most exciting era. I feel like with this new gene editing, it's very encouraging for next era, maybe next 10 to 20 years, we will have therapy in the human.
Yeah, definitely for blood cell disorders. And and the neurological disorders will take more time. There's a lot more technical issues to deal with, but
yeah, delivery, I think that would be the biggest challenge for us to decide which way to go.
Very exciting. Good to see you, Wenzhen.
Thank you, Mark. Yeah, very nice to see you. And
say hi to your husband, Jihong.
Yeah, I will. Thank you so much, Mark. It's nice to see you.
Okay, Wenzhen.
Yeah.
Bye.
Bye.